Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also often invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M, or, in special cases like today's episode, at places other than A&M. In this episode, we'll be talking to Dr. Adam Phillippy. Dr. Phillippy is the head of the Genome Informatics Section and the director of the Center for Genomics and Data Science Research at the National Human Genome Research Institute, which is part of the NIH. His research focuses on developing computational tools for genomic analyses, such as genome assembly. His team has developed many widely used bioinformatic tools and assembled the genomes of diverse species. And in 2022, Dr. Phillippy and his group published the first fully complete, or telomere to telomere, human genome. So my name is Emmarie, and I'll be one of your hosts today.
And my name is Serina, and I'll be your other host today. Well, welcome to the show, Dr. Phillippy. We're so happy to have you here.
Thank you all. Happy to be here. Blue jeans and boots. It's appropriate.
Blue jeans and boots.
I'm wearing blue jeans and boots as we speak because the weather up here in Maryland isn't the best.
I'm sorry to hear that, but I'm very glad that you are on point for this interview. It's a bummer that we are not in person so that we could see the blue jeans and boots, but I believe you.
They're not Texas boots, but they'll have to do.
That's okay. We accept it. So the first thing I have to ask you about, obviously Emmarie included in your introduction that you were part of the Human Genome Project, and specifically you helped with the telomere to telomere assembly of the human genome, the first one. So for people that are listening who maybe don't know a ton about that project, can you tell us a little overview of what that looked like for you?
Absolutely. An important distinction is kind of in the two projects. There was the Human Genome Project, which was this large international multi-billion-dollar project that was carried out in the late 90s and finished in the early 2000s, led by Francis Collins, Eric Lander, and many others. And then there was a period of about 15 years where not a whole lot happened because we had the first draft of a human genome. It was very high quality. It was done by all of the world's leading experts, and people were very happy with it through the 2000s into the 2010s. But the sequencing technology in that intervening era turned into kind of Illumina-style short-read sequencing where you would get 100, 200 bases at a time in a sequencing read. And that wasn't really the right technology to improve the genome. All we could do was measure small variants between different individuals. And as the technology improved and we realized it was possible to finally improve upon the Human Genome Project's effort, Karen Miga and myself, Karen being at University of California, Santa Cruz, put together this telomere to telomere consortium with the idea of finishing what was left, which turned out to be about 8%. And it was more of a grassroots consortium started by just Karen and myself, and we gradually added more and more people and expertise to that project over time. And in 2020, 2021, we were finally able to put the finishing touches on it and complete the remaining gaps in the human genome.
Can I ask, what was your sort of inspiration? Was it more like a need because you didn't feel like the human genome that was completed before was really fulfilling the role it needed to anymore? Or was it just like let's see if we can actually pull off creating the entire thing? Or where did it start for you guys?
Yeah, it was a very personal project for the both of us. So for Karen, her scientific interest at the time, and continues to be, was on satellite DNAs. And these are particular types of tandem repeats in the genome that repeat over and over again in a tandem array and can stretch for millions of bases at a time. And one of their primary functions and importance in the human genome is they make up the bit of sequence that the centromeres assemble at within human chromosomes. And as a satellite DNA biologist, Karen was really frustrated by the fact that none of these sequences were present in the human reference genome. So it really hindered her ability to do experimental work and to understand the form and function of these satellites. And so that was the angle she was coming from, where she really needed these sequences to do the science that she wanted to do. I was coming from it from also a personal direction, but more, I would say, of an engineering direction, where I had started in bioinformatics in the early 2000s, right at the end of the Human Genome Project. And so you can imagine, being an undergrad and having some internships in the field, and I was being exposed to this amazing project that just was all over the news at the time. It was a very impressionable moment for me. And that kind of guided the rest of my career in working with genomes and wanting to understand them and being frustrated, yes, that it wasn't complete. And, having kind of a front row seat for the first go around, I understood that it wasn't complete and why it wasn't complete. And so when the technology improved to the point that we could actually finish these difficult satellite repeats in other regions of the genome, I kind of was at the right place at the right time. I knew what needed to be done. I knew what technology could do it. And I approached it really as this engineering challenge of there's these last bits, they're the hardest. It's really bothering me that they were never finished. Let's go do it. And it was around that realization that Karen and I teamed up and realized that we had these complementary interests and strengths, that Karen really knew the biology. I didn't know what was in these gaps. Karen did. And I knew how to build a team that would help do the technical aspects of finishing it off.
So I guess that raises the question is what initially drew you to the field of bioinformatics?
Yeah, it's a very simple answer. It's the name of a professor, Dr. Art Delcher, who was a computer science professor of mine while I was an undergrad at Loyola University, Maryland. And I went to undergrad knowing I wanted to do computer science because I enjoyed programming and I was good at it. But I had no idea I was ever going to do bioinformatics. I never really took biology courses or life science courses. I was more of kind of a math, physics, computer science type. But when I was sitting in one of Dr. Delcher's classes, he announced that he had just received a research grant to do some software development. And he had invented one of the early gene finders, a tool called Glimmer. And it was an HMM-based model for finding genes in bacterial genomes. He had received funding, I think, from NIH to continue its development, and he needed an intern or a research assistant to help him with that. And I volunteered, and he took me on and mentored me for probably 3 out of my 4 years of undergrad. And I realized that applying my computer science skills to these really interesting genomic questions was a really great match, because the genome is just a string of characters in the end. And it's really easy to get into the field if you're a computer scientist because it's just text files and you know how to read in and process strings of text. And so it was kind of an easy introduction to the field. He gave me the basics of what I needed to know on the genomics and biology side. And then it just went from there.
That's really cool to hear you say, because as a biologist who is during my PhD having to learn how to use computers in an effective way to do the biology I want to do, I know from that side of things, it's been a learning curve for me, at least. Do you feel like there was a learning curve in terms of the biology for you, or was it relatively easy to pick up because you had the computational background and that was more prevalent, I guess, in your work?
Oh yeah, I'm on the 24th year of my learning curve when it comes to genomics and evolution and biology. It's something that I've really enjoyed. I was never drawn to bio classes in school. I never had a bio class in college. I had maybe one kind of intro to biology in high school. I never took AP Bio or anything like this because I really didn't enjoy the memorization components of it. Mm-hmm. I would have liked the lab components and so forth, but memorization I'm not good at, and I never enjoyed. I like problem solving. Mm-hmm. But this on-the-job training over the past 24 years in genomics, I've really enjoyed because I get to learn what I'm curious about. I don't need to worry about memorizing stuff and taking tests. I can just Google whatever I need to know. I can read the papers I'm curious about. And you learn what you need to learn to solve the questions that you're asking. And that part has been really gratifying. And so I've taken 24 years of a genomics class now in the real world. And that's been really fun and gratifying. And I would say I learned a lot more over those 24 years in genomics, and I grew than I did in computer science. I've definitely grown more into the genomics than I did the theory or the computation or the math. And I'm still using the stuff I learned in undergrad and grad school on the computational side.
How would you say that, genomics or bioinformatics as a field has evolved during your 24 years plus in it?
Oh, wow. It's evolved
Big question.
Tremendously, and it's evolved very rapidly. It's, I would say it's an exponentially growing field for the past 20 years and continues to be so. And I think the growth has happened both on the computational side and on the biochem engineering side. And so on the computational side, everybody knows Moore's law. Everybody knows that the amount of storage has just continued to increase exponentially. Our ability to compute and parallel computing has continued to increase. And so all of the stuff that we did now in the telomere to telomere project would not have been possible 20 years ago, because the scale of the data is bigger than we could have possibly stored. And the algorithms that we're using to compute on that data, namely the machine learning methods that are used to do base calling, to actually guess which ACGs and Ts are going through the nanopore, for instance, those are all based on modern machine learning techniques that weren't available 20 years ago. And they're done on GPUs of the type weren't available 20 years ago. And so it's this really interesting mesh of the computer science, the algorithms, the computer engineering, the biochemistry, the physics, and everything together is this hugely interdisciplinary field that have made sequencing possible at the level it's done today. And all of those advances were interdependent on each other. And so it's really fun to be an interdisciplinary scientist and have a really clear view of all of the pieces that went in to make this technology possible. And it really is mind-blowing, even for somebody like me.
Are there any sort of emerging technologies or methodologies within bioinformatics that you foresee being like especially useful now that we have, I guess, all these available resources?
Yeah, I think the one elephant in the room is machine learning. And so everybody should now be aware of natural language processing and large learning models like GPT-3, GPT-4. Those are true advances over the past 5 to 10 years in our ability to build machine learning models of a higher complexity than we could 20 years ago. They can learn much more complex functions, so to speak, given good training data, large training data, exhaustive training data. And as I said, in computer science, it was easy to make the transition because we're just reading strings of text. My first project in bioinformatics, it was like, here's a FASTA file. A FASTA file is really just a text file that has 4 characters in it, and it goes on for millions of bases, and this is a genome. And so I was like, great, read in text. We know how to read strings. They teach you that in the first year of computer science curriculum. Similarly, these large language models are built to just read in giant strings of text and understand the context and the meaning of that text. And so there's some pretty natural applications of those methods in genomics. And I think that will have a lot of power in the next decade. In particular, for what I like to call predictive genomics. And so, for instance, given a string of DNA, and maybe given its epigenetic state, so its methylation patterns and whatnot, maybe given the cell type and the environment that it is exposed to, predict what the expression levels are of all of the genes in that cell, given the sequence and the environment. And I think that's totally doable with current models and current experimental techniques, if you collect the right amount of data and the right type of data. And so I'm really eager to see what happens in the field over these next 10 years. People generating experimental designs that generate the right type of data to train these type of predictive models that can basically become little models of the cell, little models of the genome. And then that will allow us, I think, to do much more rapid experimentation, because if you're deciding what experiment to do or what gene to test, you can do it in silico first, and that'll give you a short list of the genes you should care about. And then you can go down that short list in rank order and kind of test the ones that the model predicts will have the biggest effect. And I think that'll make the whole research enterprise much more efficient. And one of the biological or experimental techniques that I'm really interested in now is our ability not just to read the DNA in terms of the ACGs and Ts, but we can read these single molecules of DNA through a nanopore or through a polymerase with Oxford Nanopore or Pacific Biosciences sequencing. And that gives us the ability not just to see the ACGs and Ts, but the modifications on them in terms of the methylation, in terms of even accessibility of the DNA, whether it's wrapped up with a nucleosome, whether there's transcription factor binding happening at various spots. And all of that interesting metadata layered on top of the DNA, I think will give us a lot of predictive power in these machine learning methods. So getting that very rich genomic data, epigenetic data, and feeding it into these new machine learning models, I think is the most promising avenue for the next 10 years.
Can I ask, do you think that machine learning is going to change the way that we look at medicine? So like the way that we do, like I know personalized medicine is something people are talking about a lot now. Do you think that that'll affect the way that we treat people in the future?
Yeah, I think it's inescapable that machine learning will touch everything, for good or bad, in the next couple of decades. It's the new shiny new tool. There's a lot of hype around it, but there's a lot of truth to what it's able to do. And so anywhere it can be applied, somebody's going to try to apply it for profit. We're on the NIH side going to try to apply it for good in science and research and human health. And hopefully we end up at a good place, but absolutely in precision medicine and in personalized genomics and elsewhere, I expect it'll play a leading role in the coming years.
I guess that makes me kind of curious. So given that there's been so many recent developments, both in the technology and then also like on the bioinformatics side, how do you sort of approach when you're mentoring students or like leading a team, like in terms of both like writing new tools or like identifying stuff that might be useful for your team to use?
Yeah, that's a really good question, because I even, having been in this field like 24 years now, often feel overwhelmed by the thought of like, what should we do next? Because there's so many directions that you can go in. And I particularly am feeling that now because it was kind of my career aspiration to finish the genome. And I thought it would take us a long, long time. And it went a lot faster than I expected. And now we're kind of stuck. Like, what do we do now? And so I've been thinking about this a lot lately. And you have to strike a balance of what you're curious about and what you'd like to learn, which always is kind of driving my motivation, with what you're good at. Because it is a bit of a competitive business, research or academia. And if you try to do stuff you're not good at, you will finish last. Like, you'll finish, but it'll be a hard go of it. And so you definitely want to lean into your strengths. And so I'm always looking for, kind of in myself, what am I capable of doing and what am I interested in doing? And I try to find a happy medium. But then in my people, you've got to recognize that they're all different from you, and they all have their own curiosities and their own strengths. And as a mentor, I think the best thing that I can do is, with my experience, look at a student, look at a postdoc, even a staff scientist, and kind of understand, what is the special thing that they're bringing to the table that I think that they can do a lot better than anyone else in the lab or anyone else in the field, and kind of nudge them in that direction and put them on projects where they really have a good chance of succeeding and that it's something that they're personally interested in. And so whenever we have a new summer student or a grad student enter the lab, the first couple of weeks is always a back and forth discussion of like, well, what do you like to do? What are you good at doing? What do you want to do? And we try to find projects that fit people's strengths, and find projects that are complementary with all of the people that are in the lab, because they're not in isolation, they're working with other people. So the ideal project is one where you've got multiple people that are all bringing their own unique talent to it. They're all interested in it. They can collaborate well together. And then they all learn from each other. And so one person's strength, another person's weakness, the weak person learns from the strengths of the other person. And at the end of the project, they've all grown based on everybody else. So I find that kind of pair programming or triplet programming works for projects as well. And it's the best way to train people, is that kind of on-the-job training, learning from people that have done it before. Because then you're not just starting from scratch. You're kind of learning the best practices up front, and that can save you a lot of hassle.
When you're building your team, looking for, like you said, either graduate students or postdocs, staff scientists, undergraduates, interns, anybody, what do you kind of look for in the people that you take on?
Number one is enthusiasm and eagerness and a real strong desire to learn and do good in the world, so that they have that internal motivation. I'm a very kind of hands-off mentor, and I can't be just, cheerleading constantly to get somebody across the finish line. I like people that are really self-driven, self-motivated, and are a self-starter. And if they run into a problem, they'll go try to figure it out. And by figure it out, I don't mean that they, just kind of endlessly read papers. They know to go up to somebody who knows the answer and ask them. And so that they'll come into my office when they're stuck and ask me, or they'll ask one of the more senior people in the lab for help, and really just figure out, problem solvers. They're presented with a problem and they know how to get around it and aren't afraid to figure out a solution. And then second, it's really kind of a base set of skills that you need to succeed in the type of research that my lab works on. And so we're very heavily kind of engineering and applied. We're not, theoretical on the algorithmic side. We're not like theoretical, population genetics people. We're kind of engineers. We see a problem and we want to figure out a computational solution for it. And to do that, they kind of have to have a base set of informatics skills, comfortable at the Linux command line, know a scripting language, preferentially also know a programming language like C, C++, or Rust or something like this, so that they can be efficient with these really large data sets that we deal with. Because if somebody comes in and they don't have that kind of prior scripting analysis, scripting skills, we're not a university at NIH. We're 100% a research enterprise. And so we don't have the ability to send people to classes and kind of train them up on those basic skills. So we're generally taking people a little later in their career, kind of towards the end of a PhD or a postdoc, where the universities have built those essential informatics skills and we're ready to now apply them to the problems that we're after. And so just being comfortable moving around, terabyte-sized data files is probably the most important skill for the type of work that we do.
I'm going to backtrack a little bit because we talked already about sort of the evolution of technology throughout your career. And so I'm going to ask you, and I'm biased because I really enjoyed your talk when you came here. You gave a seminar here a few months ago now. And I really enjoyed listening how you guys leveraged technology now to finish the human genome. Could you talk a little bit about that for the people listening to this podcast?
Yeah, absolutely. The one sentence answer to like what enabled the T2T genome was long read DNA sequencing.
Right.
And so we mentioned at the top of the show how for the 2000s, it was really dominated by short read sequencing, sometimes called next generation sequencing, which was important because it allowed scale like we've never seen before. So, the 1,000 Genomes Project as an example, we could actually go out and sequence thousands of human individuals and characterize a number of their differences relative to each other. Not all the types of variation, but small single nucleotide polymorphisms, small indels, and so forth. And that allowed us to operate at a scale we had never been able to do before. And that's why it was so popular, because it enabled this kind of comparative genomics across the human population. And it enabled some of the early genomics wins in healthcare, characterizing mutations associated with disease risk or certain Mendelian diseases and so forth. But to get at that last 8%, what's in that last 8% of the genome is really, really nasty repeats. And so we talked about the satellite repeats, which are just these large tandems. But another class is what we would call segmental duplications, which are not a tandem repeat per se. They can contain them within them, but overall generally not a tandem. And it's like a bulk copy paste of a bit of the genome very recently in evolutionary time to some other place in the genome. And all of the genes and whatever functional sequence is in that multi-megabase duplication comes along for the ride and gets duplicated elsewhere in the genome. The problem is, because a lot of these are very recent, on the human branch, basically, are human specific relative to the other great apes, they haven't had a lot of time to accumulate mutations. And so those duplications are very, very similar to one another, to the level of like 99.9% similar. So if you're looking at a 100 or 200 base sequencing read, there's no way to know which copy of that duplication it came from, because there's no uniquely identifying SNPs or variants within it. And so you're just stuck not knowing where it came from. So, imagine doing the jigsaw puzzle and there's two identical copies of Waldo in the jigsaw puzzle, and you're looking at a picture and it's got the little stripy shirt on from Where's Waldo, but you don't know which of the Waldos it's from. Same situation. The long read sequencing technologies came along and boosted the length of these sequencing reads, so the length of a bit of DNA that we can read at one time, to originally like 10,000 bases with the PacBio instruments, and now with Nanopore over 1 million bases. And so this just made our puzzle pieces much, much larger. It included all this additional context. And so now when you're looking at a repeat and you have a 1 million base pair read that's originating from it, there's a near certain probability that within that 1 million bases, there's some unique variant that's specific to that repeat copy. And so it lets you uniquely place that read on the genome and say, okay, I know where this came from. It came from this part of the puzzle. And with that extra context of these long reads, it allows you to build the whole genome back again and complete the puzzle. And so it was just very simply those much larger puzzle pieces that let us put the human genome puzzle back together again and get all of the repeats sorted out. And now the important next step is that for all of the functional assays, for instance, RNA-seq assays, so we're measuring expression levels, or you might have heard of things like ATAC-seq that are measuring the accessibility of chromatin, Hi-C measuring the conformation of chromatin. All of these assays in the past 10 years have been developed on short read platforms. And so their readout for this is a chromatin contact is 100 base pair read. And so you run into the same problem of in the repetitive regions not knowing which part of the genome this contact came from or this transcript came from or whatnot. And so now we kind of have to shift all of the functional assays to the long read space as well, so that when we're measuring transcripts and it's from one of these pyrologous genes that are duplicated many times, we know which copy it came from. And we also have to have the genome of that individual that we're sequencing, because in many cases, these repeats will be different from human to human or even between the two haplotypes in your diploid genome. And to map that very confidently and accurately, you kind of have to have the genome of the individual that you're getting the transcripts from or doing the functional assay from. And so doing long reads and doing them on personalized genomes is the point at which we are now and the type of work that we'll be doing for the next 10 years.
So besides the Human Genome Project, which obviously is a huge contribution to the field of genomics, what would you consider is like your other most significant achievement during your career so far?
Yeah, well, definitely the Human Genome Project was the one that I would list off the bat as the one that I'm most proud of and was most significant and that will live the longest of the things that I've done. But if I were to go back on the more kind of technical informatics side to some of my kind of proudest tooling development, it would have to be some of my early work on genome alignment. And so it was really the first projects that I worked on when I was working with Art Delcher as an undergrad and then at the Institute for Genomic Research early in my career, was developing tools that could align the first genomes that we had. So this was, the human genome had just been done, but it was one genome. There was nothing else to align it to. Shortly thereafter, we had like a mouse genome. There was a fly genome. On the microbial genomics side, for instance, we had like the first two Streptococcus genomes or the first two Staphylococcus genomes. And the first question is like, how are they different? And there was no methods at the time, because we never had this problem of comparing genomes before, to line them up and understand what the differences are. And so one of the other tools that my advisor, Art Delcher, had developed was a tool called Mummer, which was a tool for finding exact matches between two genomes. And that was one of his kind of foundational contributions to the field. And one of my first projects that he tasked me with was to take these exact matches, which give you some amount of information, but limited because it's only telling you what exactly matches, and figure out the inexact matches. And so I extended Mummer into this tool called NHGRI and then Promer, which were tools designed to line up and catalog all of the differences between two complete genome sequences. And some of the methods that we developed for Mummer and NHGRI are still used to this day. The tools are still used, although there's some faster tools around, such as the Minimap aligner from Hung Lee. But even these modern aligners are still based on kind of the fundamental strategies that we developed for Mummer and NHGRI back in the day, which is like find some exact match seeds, cluster them together in some intelligent way so that you can put all of the exact matches together that belong together from the same kind of synthetic piece of the genome, and then extend alignments between those exact matches to find all of the individual SNP differences in indels using, basic kind of Smith-Waterman type approach between them. And then report out for these two genomes, here's all the bits that match, and here are all of the differences. And so fundamentally, you're trying to build what I would call an orthology between the two genomes. For like every nucleotide, you want to connect them to one another or line them up if they evolve from the same ancestral base or the same ancestral sequence. And it's a really hard problem that still gets a lot of attention today in development. But I think some of the early work that we did on that problem has had a lot of influence in the field. In particular, even on the visualization side, where we had a tool called Mummerplot that came along with Mummer that would generate these dot plots between two sequences. And now anybody that's done sequence analysis has seen a dot plot showing the alignment between two sequences. And we made some of the first visualization tools that were kind of easy to use and quick to generate these dot plots. And so, yeah, genome alignment and some of those early methods is what I would list as my other significant achievement to date.
I feel like the alignment achievement, in addition to the human genome achievement, and you mentioned the 1,000 Genomes project, kind of all coalesces well into asking you about pangenomes and what you think the future is for pangenomes, for humans or for anything, sort of moving forward genomics into pangenomics.
Yeah, absolutely. And it's absolutely true that there's connections between all of these, that the idea of building a pangenome, which is sometimes put forward as we have a collection of a bunch of human genomes and we want to understand their relationships to one another. The first step of kind of building that is aligning all of the genomes to one another and understanding their evolutionary history and understanding how they're all related and how we are all related. So yeah, absolutely. It's beautiful, full circle connection to kind of how I started and what we're working on now. I have a couple of things to say about the pangenome. The first thing that I will say is that it can sound like a complicated term or a complicated field. Sometimes people will talk about a graph. We'll talk about graph genomes. And especially clinicians working on clinical genetics might get a little scared or intimidated by these ideas like, oh, this has no application to what we're doing. This is just theoretical stuff that computer scientists are playing around with. But I like to emphasize that a pangenome is very simply a collection of human genomes. The alignments and the graphs, these are just ways of storing the data or understanding their relationships between it. But as its very most fundamental concept, a pangenome is a collection of human genomes. If we want to be a little more specific, a collection of human haplotypes, because you have two genomes in each of your cells, the haplotype inherited from your mother and the haplotype inherited from your father. And so to build the human pangenome, our first task is just to go collect and complete as many human genomes as is feasible. And that's what we're currently doing. With the same assembly methods we developed for the T2T project, we're now just replicating that across many different human genomes. And the motivation for that project is very simply to reduce bias and improve equity in genomics. Because if I sequenced my genome and I announced that my genome is the reference genome, and then forced everybody in the world to compare their genome to my genome, it's going to be easier to compare the genome of my family members or the people that are close to me in my family tree than it will be for people on the other side of the world. And because of that, certain variants that are associated with diseases might get found in the people that are more related to me more easily than people that are more distantly related to me. And so it's really just this bias that's introduced by the use of a single reference that we're trying to get rid of, so that if somebody comes into the clinic and they want to have their genome analyzed, we can compare it against all of humanity, basically. And within all of humanity, there's going to be a good match. The family tree is not that big. We'll find somebody in that collection that has a very, very similar genome to them. And then we can line it up to that genome and say, okay, what's different between this genome and the next nearest genome that we know about? And there's some caveats about recombination and all of that within the population, but it's all can be handled on the informatics side. And then the way I start to think about it is that the pangenome is really giving us prior knowledge about what a human genome typically looks like. And so if we collect a few thousand human genomes, that's just saying, all right, here's a few thousand genomes, and think of it as like a prior probability. So when you see somebody in the clinic and they're sequenced and it has a variant that you've never seen before, okay, this is a little odd. It's not in the family tree. This is some recent de novo variant. More likely that this is the cause of some unknown disease than variants that we've seen before. And that very simple kind of probabilistic reasoning based on allele frequencies has gone a long, long way. And there's databases like Nomad that some people might have heard of that basically consolidate all of this allele frequency information so that if you have a patient with a rare disease, the first place you look is all the variants you've never seen before in the population. And we've been able to do that with the 1,000 Genomes project and large exome projects and so forth. But the trick now with the Pangenome project is we're doing it not just for small variants, but for large structural variants. And we're doing it for the whole genome. So we'll have this really complete, comprehensive view of what a human genome looks like. So now when we see a structural variation, we can also guess, how frequent is this? How likely is this associated with disease? And then it goes back to the machine learning models we were talking about earlier, with all of that prior information of what a human genome typically looks like. And hopefully we'll have metadata on the clinical outcomes of those genomes. You can train models that, given like every base of a genome, hopefully could output some kind of risk score for certain diseases or point you in the right position to look if you're trying to diagnose a previously undiagnosed disease. And so just getting that very, very basic fundamental information of what human genomes look like is the whole focus of the Pangenome project. And that'll have a tremendous payoff, both for general science and in the clinic for health outcomes.
Moving away from professional and a little bit more into like personal insights and balance, given that you work on these very large, very complex, lots of moving parts projects, how do you sort of maintain like a work-life balance? And what does that look like for you, I guess?
Well, telework definitely helped over the pandemic. The majority of the informatic T2T projects happened in the summer of 2020 when everybody was on lockdown. And so we weren't traveling, we weren't coming into the office, we were all organized on Slack and on Zoom. We were all working from home. So I have 2 daughters and both of them were at home at the time. And they loved it because I wasn't traveling anymore and Dad was just around. So anytime they wanted me, even if I was working, they could just walk into my home office and do whatever. So it gave me a lot of flexibility. That's the one thing I really like about this job of kind of academia and science. You have a lot of work flexibility. So you can work when you have the time to work. You can work odd hours. You can work late into the night if you want. You can work in the mornings if you want. And you can work around schedules. So that kind of flexibility, I think, goes a long way to maintaining the work-life balance. But second, you have to really love what you do. It's if it feels like a job to you, it's probably not the right way to go. The curiosity and the discovery and everything should motivate you to the point that you wake up in the morning wanting to go into the lab or wanting to sit at your laptop and ask some questions or solve some problems, so that you're motivated to put in the hard work, because it takes hard work. Absolutely. Like for the T2T project, I never worked harder in my life. And there were some days where I would just wake up and work all day long and then go to sleep. But it comes in spits and starts, that there'll be a set of days in a row where you just get in a flow and you just are working nonstop. And then you can take the next 3 days off and do almost nothing, is how I find myself operating. And so that flexibility is nice. The ability, once you get in the flow, to stay in the flow and really just do some really, really hard work is nice. But you have to like it, because it is, like I said, a competitive business. Research, people are competing for funding. People are competing for positions. Sometimes they're competing to, beat somebody to a discovery. And if you're not in it and willing to do hard work, somebody else will probably beat you. And so you do have to love it and want to put in the hard work, because nothing comes for free. I say this to my kids, there's anything good comes with hard work. It's not just going to fall into your lap. So that's where enjoying what you do is really important for me, so that it doesn't feel always like a job.
Definitely. On those days where you work really, really, really hard and then maybe you take the next day or 2 off, are there any things in particular that you use to sort of reset, any hobbies or?
I'm definitely a big fan of getting outside, go for a hike in the woods, play some sports, be active. I'm a bit of a generalist. I try a whole bunch of different things and then I get bored of them and then I'll go try something else. So there's not one thing that I've been committed to my whole life, like the genome has been. But I like sports. Right now I'm playing a lot of pickleball like an old man. My younger daughter likes to play lacrosse, so I'm in the backyard throwing lacrosse with her, or going to her games. During the pandemic, my older daughter was really into horses, and so we were out at the barn all the time. And I learned more than I would ever imagined I would have learned about horses. And I mention it because it's like one of these examples of like, I just like to learn stuff about the world that I wouldn't have thought I would get the chance to learn. And so like all of a sudden, in 2020, 2021, in parallel to this genome project, I was like becoming, an equestrian dad and learning all this stuff about horse care and veterinary care. And it was really fun just because it's something new. And I always like to learn new stuff. And so, those types of things that just get you away from your desk and take your mind off of what you've been thinking about and let you think about something new. And spending time with my family is what gives me the best decompression effect.
Dr. Phillippy, you thought it was going to take longer to do the T2T project, but COVID kept you at your computer working on it. Yeah. We can thank COVID for the rapidity
I'm honestly
of that
not
project.
sure
That's
it would have happened as quickly if it hadn't been for that, because it was really easy to build this big, across the nation international consortium that were live on Slack and Zoom, throughout the week working on this, because especially on the bio side, my group didn't have the expertise at the beginning to know what we were looking at or to know the interesting questions to ask. And so we learned so much from the people that were in the T2T consortium. And I like to think they learned a lot from us too, in this kind of mutually beneficial collaborative environment that just worked really, really well remotely at the time. Because thankfully, we had collected all the data. It was complete happenstance that like the last of the nanopore and the last of the PacBio data had come off the instruments in 2019. And so we were just sitting on this pile of data ready to analyze it. And we had the right people at the right place at the right time to sit down and work on it. In particular, in my lab, Sergey Nurk, who wrote the Spades Assembler, co-wrote the Spades Assembler, if people are familiar with that. Sergey Korin, who's worked with me for the last 14 years on genome assembly problems. Adang Ri, who led the validation and the polishing work, and most recently the human chromosome Y finishing efforts. And Mikko Rautiainen, who developed the graph construction techniques that we used in the project and assembled some of the hardest bits of the genome, like the rDNAs and the satellites. And that's just in my lab. And I had all of those people, sitting in their full attention on this project. And they are absolutely the world's experts in everything that they do. And they were all in the same place at the same time. So I think I was just incredibly lucky that I had them all in my lab at the same time. And then to pair up with people like Karen Miga, Rachel O'Neill, Evan Eichler, Winston Timp, it's just, we were so lucky to get the attention of these world's experts. And I think it was really down to the fact that we were really, really open about what we were doing. And so we put all of the data up on the web unrestricted as soon as it was generated. We were very loud and vocal. Hey, we're trying to finish the genome. Join us if you're interested. And people understood the significance of the project, and they appreciated how open we were being about it, and not at all kind of, trying to seek fame and glory. We just wanted to get it done. And we were very open about that. And that made people want to work with us. And so the consortium built itself very naturally. People just would come up to us and volunteer their time and effort. And the same with the students and the graduate students and the postdocs. And so, building that really healthy, collaborative environment that's putting the science and the people before, the fame and fortune really was essential to the success of this project. And then we got lucky that we got the fame and fortune, even though we didn't ask for it. So it was an odd couple of years after, going to award ceremonies and tuxedos and things like that none of us had prepared for.
Yeah. You talked about how research is competitive, so it's really nice that you guys didn't feel like this weird sort of competition that prohibited you from being able to find collaborators that were helpful to you. Like, you were open to collaboration. I feel like that makes science stronger in pretty much every field.
Yeah, I would say the thing that I'm most proud of looking back at the project is countless number of students and postdocs that were members of the consortium have said to me that it changed the way that they think about science. So they had seen a lot of negative press on, Twitter when Twitter was around, and now probably on TikTok about people just being disenchanted with science and research and academia. That this project changed their view of that for the better. That they're like, I realized that I can do science the way I want to do it in a collaborative, friendly atmosphere where everybody's lifted up and the science is first. And that made Karen and I so happy that we had that impact on the trainees that we got to work with, and made them go forth in their careers realizing that you can do it in a way that not only gets the results that you want, but in a way that benefits everybody in a fair and uplifting manner. You just have to have leadership of the research and of the consortium that values that. And Karen and I absolutely do. And it works. Like I've run my whole career this way of trying not, it's really hard. Everybody has an ego to some extent, and everybody kind of wants credit, and you can feel slighted if you don't feel like you're getting the credit. These are all natural emotions to feel, but you kind of have to just realize that they're the wrong emotions to feel. They're counterproductive. And so you have to push them down and say, no. Like, what's best for the science? And so if I'm trying to make a decision, it's not what's going to get me the most credit or, what's going to help me in the near term. Because often you have a very short-sighted view of that. It's hard to understand the positive things that happen if you're a nice, friendly, collaborative person. And so if you put the science first and you put your people first and you make a decision, all right, I have to solve this problem. Well, I know so-and-so who's going to solve it a lot faster than I'm going to. I'm just going to tell them about the problem, give them everything that they need, and support them and let them go solve the problem. Like, I don't need the credit for this. I just want to get the work done. And just be very open about it, and communicate frequently and openly and honestly with people. It really has a tremendous positive effect in my experience. That then the right people want to work with you, like-minded people. They will reciprocate and be very open and generous towards you. And so for me, at least, survivor bias caveats all around, it worked out. And so I definitely try to preach that to my trainees as well, that this kind of open, collaborative atmosphere has a good benefit. And I think the field has changed a little bit in the past couple of decades. And I have some of my more senior mentors will remark to me that they have watched the field change, and it's definitely become more collaborative over the years. Whereas, 20, 30 years ago, maybe it was a little more focused on one person, trying to do it or get the credit. Now, the world has changed with technology and Slack and Zoom and Twitter. We're all much, much more connected. And so I think the research also happens in a similar way. And so let's do it in a friendly, happy, collaborative way, because there's nothing worse than going to work and not enjoying what you're doing. But I'm so lucky every day I get to work with tremendous people that I just love to work with and just have a really positive atmosphere around our research.
Well, and obviously you did acquire plenty of fame from all of this because you got invited onto the Blue Jeans and Boots podcast. Extremely big deal.
What's better?
You made it.
Yeah, real pleasure to be here and chatting with you all this morning. It's always my favorite part to get invited by students and postdocs and researchers. Like I said, we all have a little bit of an ego, so it makes me feel good, giving people advice.
Yeah.
Because Lord knows my kids don't listen to it.
What would you say? We've talked about collaboration and how important it is. Is that the biggest piece of advice you think you'd give to people, your trainees, or to anybody listening to this who's maybe thinking about graduate school or a career in science?
Yeah, I do usually, one of the things I always remember to tell them is to find people to work with that you enjoy working with. Because I think a lot of times, if you're in science, you're kind of focused on the science, and I'm going to go work for the lab that's the best at this particular technique that I want to learn. I would push back on that a little bit and say, at least for me, I've really worked with advisors that I knew would support me and cared about me, put me first, and were just a pleasure to work with, right? Because there's this social aspect to doing science it's inescapable. You're always interacting with people, you're learning from people, you're training people, you're teaching them. So work with people that you want to work with. And so I always tell that to my students when they're graduating, looking for like a PhD lab, looking for a postdoc lab, like put the people first, because that'll really affect how much you enjoy your job. And what kind of people, look for people that will support you. And so as an example, my PhD advisor was Steve Salzberg, who's now at Hopkins. Tremendously loyal to his people, tremendously supportive. I had my first daughter the semester before I started my PhD. And so I was a bit of oddball, compared to some of my fellow students, going to class and I've got a newborn at home. I know there's other people that have been through this, and probably some of your colleagues as well. But Stephen was so supportive of the situation that I was in, being a new dad and knowing that I had to support my family and whatnot. He went out of his way in the summers to kind of convert me through some administrative mechanism to basically a full-time employee and pay me as a software engineer through the summers so that I could make enough money to help my family out. And then during the year when I was in class and even doing research with him, I also worked part-time 20 hours a week for somebody completely different than him. So I was moonlighting. And in the past, this was not allowed. A lot of universities might still not allow you to work outside. But Stephen was totally fine with it. He's like, do what you need to do. You've got to support your family. And so I worked as a software engineer all through my PhD, and Stephen supported me and paid me as much as he possibly could through the university. And it's just those little extra efforts that he didn't need to do, but he did it because he cared about me, that just makes everything better. And so, I just try to pay that back now to my people.
Well, thank you so much for talking to us this morning and for all of the excellent advice. I think there's lots of great takeaways from this episode, technologically, scientifically, and also just for life skills. So thank you so much for being here with us today.
Absolutely. It's my pleasure. Thanks for the great questions, and best of luck to all of you in your future research careers.
Thank you.
I'm sure you'll go on to have very gratifying successes and occasional failures, but the successes will outweigh the failures.
A healthy combination.
Absolutely. It's can't avoid it.
Today's episode was hosted by Emmarie Alexander and Serina DeSalvio and produced by Aaron DeSalvio. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, Twitter, and TikTok at Genetics Podcast. Thank you very much for listening, and we'll see you next time.